First, I want to introduce the project team because I came to this project a little bit late in the game. It was actually started by my colleague Rachel Negley, who moved her lab from here in Parlier recently to USDA ARS in Michigan, and then also Leonardo de la Fuente at Auburn University. So majority of the initial work on this project was all them, and I started in on this within the last year or so. And then I'd also like to acknowledge Rodrigo Almeida at UC Berkeley for providing all of the bacterial strains that we're using in this project. And then also my colleague in Parlier, Rodrigo Krugner, for helping maintain our field experiments. The isolates of Xylella that I'm talking about in this project, these were collected by Rodrigo Almeida's group from Pierce's disease affected grapevines all over California. And they did this as part of a very large sequencing project where they collected over 100 different isolates of Xylella and they sequenced the genomes of all of them. And what they found in the genomic analysis was that isolates from the same geographic location in general would cluster together genetically. And I'm not really going to go into very much of those comparisons because this is work that's all published if you're interested by their group. But this is where these isolates are coming from. And so these came from that collection, and we're looking at differences between isolates that were obtained from these different regions in California. The objectives of this project was first to evaluate the virulence or the severity of the disease caused by these different isolates, to see if there was anything different about them and to see if different isolates from the same area or from different areas would cause differences in disease severity. And then secondly, we wanted to see if the known sources of resistance to Pierce's disease, the resistant grape material, would hold up against a wider range of Xylella strains that had been tested previously. We did this using 3 different experimental approaches. So first, we were screening a larger number of strains using tobacco as a model system so that we were able to do more of the strains than we would have in grapevine. Then we also took a subset and we screened them in potted grapevines. And then within the past year or two, we've also moved on to inoculating plants out in the field to look at virulence differences in field-grown grapevines. First, I'm going to start with the tobacco experiments, and these were all done by Leonardo de la Fuente's group at Auburn University. And the reason we can use tobacco as a model system for Xylella infection is because you see very similar symptoms to what you might see in other hosts, such as the scorching on the leaves. And so you can evaluate disease based on symptoms and disease rating similar to what you would in other hosts. The benefit here is that these inoculation experiments can be done in a much shorter period of time in tobacco because it grows much faster. And then it doesn't require as much space or resources as using grapevine. So we use this to screen, I believe, over up to 60 isolates, different isolates for disease symptoms in tobacco. This is some of the results from tobacco, and these are disease ratings based on symptoms, so the leaf scorching. And the results are grouped based on the location where the isolate came from. So you can see there's a group that was isolated from Santa Barbara, from Bakersfield, and then also from Napa. And they used 2 reference strains in these experiments. One was a Temecula isolate, which was originally from California many years ago, that's been published in the literature a fair amount. And then also WM1, which is an isolate that came from grapevine in the southeastern US. So these 2 reference strains were included for comparison purposes. And we're able to screen quite a number of isolates this way. And what you can see is that the disease severity caused by some of these different isolates is less than the reference strains, and then there are some that are more than the reference strains. So in the tobacco system, at least, there seems to be some differences in virulence between these different isolates. But what's interesting is that it doesn't seem to be necessarily dependent on the location where these came from. You can have some isolates that are less virulent and some isolates that are more virulent, all from the same geographic area. These are just more of the same results. There's a group from Temecula, and then we did a group from Sonoma as well. And it's very similar. There are some isolates that are slightly less virulent than the reference strains and some that are more virulent than the reference strains. So in tobacco, it seems like there are some differences here. Next slide. So that's what we found in tobacco. There are some differences between the isolates in that system, and they don't necessarily seem to be dependent on geographic location. So as far as what makes some more or less virulent than others, it would have to be something that we would need to do a little bit more work on. Next slide. So then we moved on to looking at a subset of these isolates in potted grapevines in the greenhouse. And so these experiments were actually done in this greenhouse. So it's open to the outdoors, it's not temperature controlled, and it's a more natural environment. This was done in Cabernet, 1 cultivar, and we used 16 different Xylella isolates from different regions, as well as the Stag's Leap strain, which we used as a reference or a control. And there were some differences in the virulence based on symptoms, so scorching symptoms and Pierce's disease symptoms that we saw between the different isolates. But as many of you who have done this kind of experiment are very familiar with, you also get a fair amount of variation between plants inoculated with the same isolate. So it's a pretty variable system, a little bit more so, I think, than something like tobacco, but that's what we work with when we work with Xylella. So next slide. And these experiments were done twice. And when we compared the different isolates based on disease ratings, there were some differences, small differences between the different isolates in the disease that they caused. But there's also, again, a fair amount of variation within treatment. So plants inoculated with the same isolate, and then also between experiments. So these are 2 different experiments that one was done slightly later in the year than the other. The second experiment is on the right side, and in general, this one had less disease than the first experiment. So you do have experimental variation there. And in general, if you take that into account, there really isn't a lot of difference between the different isolates. They all do seem to cause disease, but there are a few exceptions. There are a few isolates that do seem to consistently come up as less virulent. I pointed out this one JE118 isolate, which in both experiments was significantly less virulent than the other ones. So in general, it seems like these differences, if you're looking at Grapevine, are not especially significant, but there are a few that do come out as different in repeated experiments. So the second part of this project is to look at about host resistance. So we're evaluating here resistant material to see if the resistance holds up across a number of different isolates that we're screening. So we used 3 different sources of plant resistance. So this is IAC 572, 8909, and Tampa were the resistant and/or tolerant material that we were using. And then we used Cabernet as a susceptible control in these experiments. And these were done in the greenhouse in potted vines. We used 7 different isolates of Xylella for this, again, from different locations in California. And even the resistant or tolerant material, it can get scorching during the course of the experiments. Some of the symptoms similar to Pierce's disease, but generally much less than you see in a susceptible plant. And some of this, I think, can also be caused by abiotic factors too, as these are potted vines. But we did see some scorching symptoms throughout the trial. Next, but in general, what you see is, as you would expect, the Cabernet has significantly more symptoms than the resistant material. And it seems to hold up pretty much across the isolates that we looked at. Again, there's a fair amount of variation between different experiments. So the blue and the green are two sets of experiments we did on this. And especially important in the case, I think, of the resistant material is that this is just based on symptoms. And so we still need to evaluate the bacterial populations in the plants. We have the samples that we're processing for that right now to see what's really going on as far as bacterial populations in the plant. So just a summary of the greenhouse experiments with the grapevine. It seems like that environmental variation, such as between experiments or within treatment variation, is in many cases more significant than comparisons between the different isolates. That being said, there are a few that do come out as different consistently across different experiments. And it does seem that the resistance and or tolerance of these different types of material does hold up across a wider range of isolates that we screened. Next, we wanted to do some comparison of virulence out in the field. So we have a field trial in Parlier. The first plants were inoculated in the summer of 2020. These are all Cabernet. And we inoculated these mechanical inoculations with 22 different isolates of Xylella, including strains from Bakersfield, Santa Barbara, Napa, Sonoma, and Temecula. And then we tracked the disease progression through that first season and then again through the second season, which was 2021. This is from the first season of inoculation. So these plants were inoculated in late June, and then we took samples in October to test using PCR. In general, the disease didn't progress very much in that first season. We did get some Pierce's disease symptoms, but in general, it didn't seem to be very well established at that point. There wasn't significant disease in many of the plants. And in PCR testing, about 5% of the inoculated plants tested positive. So in general, I think the disease just hadn't progressed very far at that point. But we kept the plants over the winter and we evaluated them again in the second season. We saw a lot more disease in the second season. So this is about a year after inoculation of these plants. We got significant Pierce's disease symptoms showing up starting in July. And we tracked them through the end of the season up until beginning of October, roughly. And these are just some photos of the Pierce's disease symptoms from that trial. In general, plants that had shown symptoms the previous fall did again this year. And plants that hadn't quite developed symptoms the first season did develop symptoms the second season. So these are just some more photos from 2021. And we also inoculated a second set of plants in 2021 to evaluate into next year as well. So we're doing a second round of this experiment. And these are the symptom ratings from the summer of 2021, so a year after the plants were inoculated. And in general, if you compare the different isolates, there isn't really significant differences between them given the variation, within treatment and between plants. But there are a couple that do seem to cause very reduced symptoms compared to the rest. So there was one from the Santa Barbara group, one from the Napa, and one from the Napa group that both cause very little disease at all. Again, we're repeating this experiment to make sure that this is consistent across replicates. In general, we found a lot more disease the second year. In the field experiments, again, we didn't see very much disease the first year. It came back much stronger the second year. There seems to be a fair amount of plant-to-plant variability, more so than in most cases the difference between isolates. And the second round of inoculation experiments is currently ongoing, which we're going to evaluate in the summer of 2022. And one last thing, I wanted to mention the comparison between tobacco experiments and the grape experiments. So the good news is, from the standpoint of using tobacco as a model for infection in grapevine, is that everything that causes disease in grapevine also seems to cause disease in tobacco. Comparison between the isolates is a little bit more difficult, again, because these differences don't, in most cases, seem to be large enough to come out across the variation within treatments and the variation between different experiments. So some of this is a little bit hard to compare these smaller differences, but in general, disease symptoms in tobacco do show up in everything that produces disease in grapevine. So with that, I'm going to end. So in general, what we found was that the outcomes of the infections due to these different isolates seems to be more influenced by plant factors and environmental factors necessarily than by differences between specific isolates. Most of them seem to be the same or have maybe small differences, but some do come out of our experiments as being significantly different than the others, a small number of them. And it does seem like the resistance and or tolerance of the material that we screened holds up across the different isolates. And we're still working on some aspects of this project, including looking at bacterial populations in the plants, as well as the continuation of the field experiments.